So I'm going to talk about an area that's really been my research focus over many years. And I'm going to pose the question, well, firstly, I suppose the wider context is the roles of folate and the metabolically related B vitamins that are so fundamental in human health. And the reason folate and related B vitamins are so fundamental is because they're involved in one-carbon metabolism. One-carbon metabolism really describes those reactions in which one carbon unit is transferred and utilized. So folate either donates or accepts one carbon unit. And these reactions are involved in amino acid metabolism, in methylation processes, and in the synthesis of DNA. And I'm going to be mentioning homocysteine later in the talk, and just to make a note really that homocysteine, one example of a methylation process is the remethylation of homocysteine to methionine, and when it's impaired because of low folate or B12 predominantly, then elevated homocysteine will be found in plasma, and that in itself has been the study of a major research area in itself. And just to say that although folate cofactors are required for up to 20 different human enzymes, the full-blown clinical sign of deficiency, which is megaloblastic anemia, it really reflects one metabolic step, and that's the role of folate in the synthesis of DNA. So there's an important step in DNA synthesis that requires folate, and when that's impaired, the clinical sign, megaloblastic anemia, arises where you have full overt folate deficiency. So that's the context. My talk today is really going to be delivered in terms of three questions I'm going to pose and hopefully answer. The first is to consider whether an optimal folate status can be achieved through natural food folate sources alone. And really moving directly on from that to sharing with you the evidence of beneficial health effects. So moving to maybe the health effects of either fortification or supplementation with folic acid. And we have some hard evidence and some emerging evidence in this regard. And I'm going to then finish up on the last bit, really, which is the question, is there a role for personalized nutrition, in keeping with the idea of today's session where we're going from precision nutrition from cell to society. Hopefully this talk will move through those areas, and I'll start with the more population-based stuff and end up with the more individual research. I just want to, first of all, put up a structure of the synthetic form of the vitamin I'm talking about. This is the pteroylglutamic acid, or otherwise known as folic acid. And I really just, rather than burdening you with too much chemistry, I just want to remind you that this is the synthetic vitamin, doesn't occur in nature. So if you ingest folic acid, it means you've either consumed a fortified food or you've consumed a supplement. Folates that occur in nature are similar to this structure, though not identical, and those differences have implications for the bioavailability of the vitamin and someone's response to an intervention to increase status of the vitamin. And so the folic acid molecule is comprised of three components. There's a pteridine joined to a PABA, a paraminobenzoic acid, which in turn is linked to a glutamic acid residue. Folates that occur in nature are typically reduced at the pteridine ring, so you've got extra hydrogens at 5, 6, 7, and 8 in the pteridine. And in addition, instead of being a monoglutamate, as this structure is, you've polyglutamates, so there's several additional glutamates. And these structural differences, as I said, will have big implications for how the vitamin is handled and what its bioavailability will be like in humans. And so if we just summarize that, folic acid, the synthetic vitamin, is fully oxidized. It's a monoglutamate. Folates in nature are reduced molecules, and they're predominantly polyglutamates, so they've got lots of additional, typically 7, but up to 13 additional glutamates. And all but one of these need to be removed for normal absorption of the vitamin. And that's a process that can be affected by various food factors and other factors that may limit the bioavailability of polyglutamate sources. And really the point is that the potential, the upshot of these structural differences between folic acid and the natural vitamin means the potential to influence your folate status through an intervention by means of natural food folates alone is extremely limited, because the natural vitamin can be unstable during cooking and incompletely bioavailable once it's ingested. So at 2 steps, both when you're preparing the food before you ingest it and once you have ingested it, it is it's more limiting in terms of your ability to optimize status. And so that's why studies from ourselves and others have found that it performs poorly, the natural vitamin, compared with folic acid when tested, and we'll have a look at that. So really, if you wanted to optimize folate status, if you believed in some of the health effects, and we'll be looking at those, and you wanted to optimize folate status in individuals or in humans, you could ingest natural sources of it, such as green leafy vegetables, fortified foods, or dietary supplements in tablet form. And so many years ago, we published in that journal that was mentioned a couple of times in Faithful Research Reporting this morning, the Lancet, and this was many years ago. Geraldine Cuskelly was actually my first PhD student, having moved from industry into academia. And we were interested at the time, knowing the chemistry and the biochemistry of folate, we were interested that the UK Department of Health at the time was saying folate is really important for women of childbearing age, to prevent neural tube defects, and there are 3 ways that a woman can get their folate. You can either take fortified foods, supplements, or natural foods. So at the time, the advice from the Department of Health in the UK was very much implying that these were 3 alternatives that were available. And we were interested in testing that in a trial. It was a simple trial, not complicated, but we were just comparing those 3 routes in terms of women achieving optimized folate status, women of reproductive age, to optimize their folate status. So we recruited from the university women of childbearing age who were not planning a pregnancy, so they weren't being directed specifically to take a folate supplement. And we looked at, we did an intervention. It wasn't in a metabolic unit. People were provided with their supplements, their foods, or their fortified foods on a daily basis. They were delivered to their homes, and so they were really required over a 3-month period, and the end point was red cell folate status, the best long-term marker of folate status. And we really showed that folic acid supplements and fortified foods were, fortified foods and folic acid supplements had equal ability to optimize folate status, as reflected in a significant increase in red cell folate, but increased foods naturally rich in the vitamin resulted in no significant response. And our interpretation of that was that it was due to the lower bioavailability and stability of the natural vitamin compared to folic acid. As I say, a simple study, I suppose, because we were challenging the current policy at the time. Lancet were interested and published the paper back then. Over the years, we've been looking at fortified food as a source of folate, not just in women, but in the population generally. And this was a paper from several years ago published in AJCN. And here, this was an observational study rather than an intervention study. But here we also looked at really fortified foods as a source of folate intake and status. In typical adults, healthy adults, we simply recruited up to 500 of them, studied in detail their dietary intakes by a combined food frequency questionnaire and a diet history method, and we measured biomarkers for folate and the related B vitamins. And here I'm just showing homocysteine and red cell folate and serum folate, which is another marker used for folate status. Not as good a long-term measure, but nonetheless probably used more in more studies than red cell folate levels are. And again, we looked, we divided the sample into those that reported consuming fortified foods. In Ireland and in the UK, fortified foods are available to consumers on a voluntary basis as opposed to mandatory fortification. So consumers can either choose a fortified food or not. And so we found that about a quarter of the sample were not consuming fortified foods, whereas 75% of these consumers, these regular local participants recruited in Northern Ireland, about 75% of them were consuming fortified food, and we used the folic acid content in the fortified food to divide them into low consumers, medium and high consumers of fortified food. The point really of this slide is to show that although these are observational data, so it does come with the health warnings that we heard in this morning's session, but just to say, when we looked in detail at the dietary intake and split their dietary consumption into added folic acid and natural folate, I should say that these people were all recruited on the basis of not taking a supplement. So the only folic acid coming into their diet would have been via fortified food. And you can see that added folic acid goes from 0 in the non-consumers to about 25 micrograms a day, 60 micrograms a day, and in the highest group, over 200 micrograms a day. Natural food folate didn't change, so we can assume reasonably that the increases we saw in the biomarkers were owing to the added folic acid. Natural folate was also measured and didn't change across the 4 categories, but what we showed was that red cell folate increased in an incremental way as you went from 0 to high consumers of folic acid, and correspondingly homocysteine levels decreased. These were not population-based data, and more recently then we've had the opportunity through a large study that has just come to completion in Ireland, the Jingo project, which was funded by the Irish Department of Agriculture and had 3 component studies, one of which was a national survey in Ireland, the National Adult Nutrition Survey. And unlike previous surveys in Ireland, which only looked at dietary intake, the NANS survey also took a blood sample. So as well as measuring the dietary intakes of Irish adults in detail, biomarkers of nutrient status were also measured, giving us the opportunity to look at red cell folate levels according to whether Irish consumers were non-consumers of fortified food or supplement, consumers of fortified foods but not supplement only, and then the group that consumed both fortified foods and supplements. So again, reminding you that folic acid only arises in the diet through fortified foods or supplements. So you're going from a situation of 0 folic acid intake to folic acid intake almost 300 micrograms a day, which was coming from the combination of fortified foods and supplements. And you can see that the biomarker levels increased very significantly across the categories. And these data are important because the Irish government in the last few years, like the UK government, has decided, after much deliberation and many years of consideration, has decided that we don't need mandatory fortification with folic acid to prevent neural tube defects. We would have been the first 2 countries in Europe to do so, compared to the US and Canada and many parts of the world. The Irish government were considering this and really found that because of some published papers in the area of a concern regarding folic acid and cancer risk, both the UK and Irish governments decided not to fortify with folic acid on a mandatory basis. And the idea was, really, we have a voluntary policy, and most people are getting enough folic acid because of a voluntary policy. And I would say that the danger of that is that people are getting false comfort from mean intakes. So for sure, people who are consuming fortified foods or supplements or a combination of both don't look like there's too big a concern, although fortified foods alone just puts them below the level. If it was a woman, these are average data, but a woman with a red cell level of 885 nanomole per litre is just below the threshold that we consider maximally protects against neural tube defects. But certainly there's no question that those who fall into the category of choosing not to take a supplement or fortified foods, about 20% of all Irish adults, there would be concern if a woman was in this category and had an unplanned pregnancy, wasn't taking a supplement, then there would be concern about the risk of neural tube defects. I'm reminding you that Ireland has one of the highest risks in the world of neural tube defects. So just pointing out there, those are the categories of fortified food. Really, just to say then, looking at another country, I said that in Europe, in UK and Ireland, fortification is allowed on a voluntary basis, and it would be allowed in other countries on a voluntary basis. But fortification policy varies quite considerably throughout the EU, going from countries like the UK and Ireland that allow fortification fairly liberally to other countries such as Denmark that don't permit the sale of foods that are fortified at normal retail outlets. And really, and the view would be that Danish people are eating a healthy enough diet, and they also know when to supplement, and women planning a pregnancy will always supplement, and that would be the reasons that are given for not allowing fortified foods to be sold in Denmark. But you can really see here that these using the cut-off of this is a clinical cut-off using serum folate values, a clinical cut-off of deficiency, and just data from a considerable number of a big sample of over 6,000 Danish adults. This wasn't a population-based sample, it was just an opportunistic people coming into hospital in Denmark, being referred into a hospital clinic, and they measured folate status. And really, there was a very high prevalence of what would be subclinical deficiency, and even major portion of the Danish population with levels of folic acid that would be considered near clinical deficiency. And this is a country that doesn't have a problem with malnutrition, for example. So just showing you that in the absence, that in countries that choose to fortify or have available fortified foods that consumers can choose, really they deal with a big problem that's potentially there in terms of folate status in otherwise healthy populations. So to summarize on this part of the talk, if you just look at the various strategies one has to optimize folate status in individuals and in populations, really by now, hopefully I've shown you that at an individual level, natural food folates don't work in individuals in terms of optimizing folate status by way of an intervention, so unlikely to ever work in a population level, even with an education program that advises people about sources of folate. Supplementation works very well in those who are given supplementation, but there are problems translating that to population-based preventive strategies, and I'll just talk about that in terms of the health effects of folate. Folic acid fortification works in individuals who are given fortification, who choose to consume them, or on a trial where they're provided them, and also, as we've seen from evidence, again, I'll also show this evidence in the context of the health effects that I'm moving on to now. Nowadays, as a result of mandatory fortification that started in North America, nowadays has been extended to over 70 countries in the world. So there are over 70 countries in the world that fortify with folic acid. And really just to say that this is a map that talks about fortified flour with both iron and folic acid. So there's a few exceptions, just to say Australia, which doesn't actually fortify with iron, fortifies with folic acid only, and Venezuela, the United Kingdom, the Philippines, and Trinidad and Tobago that fortify with iron but not folic acid. All others, it's a combination of both. But you can see clearly a big gap in Europe in terms of no fortification policy. The UK looks like it's filled in, but that's iron only and not folic acid. In terms of preventing neural tube defects, this audience will well be aware of the important role folate plays in preventing neural tube defects. And as a result of conclusive evidence published many years ago, throughout the world, there's clear-cut advice for women planning a pregnancy. For recurrence of NTD, women are advised to take 4 milligram a day folic acid. For the prevention of first occurrence, it's a tenth of that, 0.4 milligram or 400 microgram. And really, we've been looking more recently at the uptake of this advice by women of reproductive age. And we just looked locally in a local hospital and sampled women at 14 weeks of pregnancy. Now, by this stage, they were supposed to have been on folic acid from preconception. So the good news was that 84% of these women at 14 weeks of pregnancy were taking folic acid, but only 1 in 5 had commenced folic acid before conception as recommended. So only 1 in 5 were following the recommendation as they were supposed to be following. And that's really important because the neural tube is such an early event. The closure of the neural tube closes between 21 and 28 days after conception. And this is a time when most women are only beginning to suspect that they might be pregnant. So really starting to take folic acid when the pregnancy is well established is just simply too late. We looked at the blood levels of these women at 14 weeks, and not surprisingly, red cell folate. We asked them at 14 weeks how long they'd been taking folic acid, and obviously folic acid was highest in the women that started preconception as recommended. Intermediate levels were found in those that started sometime between 0 and 6 weeks post-conception, so around the time that the neural tube was closing. And then lowest levels were found in those who really only started taking folic acid after week 6 of pregnancy, which was far too late. And that in turn could be, those data could also be examined in terms of the cutoff red cell, maternal red cell level that's associated with lowest risk of NTD. So really, you can consider optimal folate status in terms of a red cell level that maximally protects a mother against a neural tube defect. And that obviously greatest proportion of women who started folic acid late on had a red cell level at 14 weeks that would have not been protecting them at all. More than 50% of them had not achieved a red cell level that would be associated with the lowest risk. You might be saying, but sampling at 14 weeks, what would that have to do with neural tube closure? The red cell measure gives you a bit of a history of the red cell status in that woman. So previous 3 months, so it would go back to the time, the critical time of neural tube closure, so it's important. And really, these data from published a few years ago, or nearly 10 years ago in the BMJ, really showed that, not surprisingly, these are 6 countries shown, but really the paper describes 13 countries, and really what this paper shows is the really ineffectiveness of the advice to women to take a supplement across 13 different European countries. So in the absence of mandatory fortification, simply telling women planning a pregnancy, as they do in Europe, to take folic acid simply is making no difference in terms of NTD risk. There's been no change in NTD prevalence across 13 countries before and after implementation of the advice to take a supplement preconception, because the women aren't taking it on time, presumably. So really, in contrast then, what evidence do we have the fortification works? Mandatory fortification has been in place for several years in a number of countries worldwide, and this measure has reduced the instance of neural tube defects by up to 50% in Canada, varying somewhere between 27% and 50% reduction. And as mentioned, by contrast, there's been no change in NTD rates in Europe since the introduction of these recommendations. They're in place for over 20 years, and there's been no change in NTD as a result. So really, I'm going to very briefly mention the last part of my talk in relation to the personalized nutrition concept. And really, I'm going to talk a little bit about an area that, as you look at the areas of health that folate is linked to, from maternal health and fetal development, where the evidence is conclusive, right through to conditions of aging, where the evidence is still emerging to say for sure. But I want to spend a little bit of time on stroke and heart disease, and really to show you that folate is involved in homocysteine metabolism, as mentioned, but so too are 3 other B vitamins, methylcobalamin, otherwise known as vitamin B12, and pyridoxal phosphate, otherwise known as vitamin B6. And we've been looking at these nutrients and their interaction with genetic factors. And I'm going to move swiftly on because of time, to talking about a polymorphism involved in MTHFR, this step in folate recycling. So a polymorphism in the gene, MTHFR gene, and bearing in mind that it requires FAD, otherwise known as riboflavin, as its cofactor. And so I'm going to just bypass that, but just while we're whizzing through, to say to you that a lot of people write off the fact that folic acid may or may not have a role in stroke. And this interesting meta-analysis shows that if you look beneath the evidence, and this meta-analysis of randomized trials showed that folic acid certainly had a protective effect against stroke when you look specifically at trials of longer duration or in people that had no previous history of stroke. So sometimes, both in the trials and in the meta-analysis, we talked about how you don't get the expected results. Sometimes it's because the trial hasn't been targeted at the right people. Many trials on folate and the related vitamins were targeted at people that had optimal status to begin with, and perhaps therefore never showed a response, or indeed had established disease, already had a stroke, so giving folic acid doesn't prevent another stroke. But I want to move on, and some evidence from the US population that there's been a decline in stroke mortality that supports the view that this coincides with mandatory fortification, perhaps offering some bit of support for the view that folic acid might have had a role in stroke prevention. So I want to talk about this polymorphism. It's of interest. It's been linked with increased risk of cardiovascular disease. People with the homozygous TT genotype for this polymorphism have anywhere between 14 to 21% excess risk of CVD, but major geographical variations. So following on from Dr. Seisel's point that really it depends on the population and the nutrient status of the population. We're looking at whether the phenotype is there and therefore the disease risk. So if we just summarize the polymorphism, the substrate is one folate cofactor, 5,10-methylene THF, the product is 5-methyl THF, the cofactor is FAD, and the precursor for that cofactor is riboflavin. And a number of polymorphic mutations have been described on the MTHFR gene, but the one that's most studied and the one of greatest interest certainly to ourselves and most researchers in the area is the C677T polymorphism. So it involves a C to T substitution at base pair 677, an alanine to valine change in the amino acid, and a functionally defective enzyme. We initially looked to see homocysteine change, and we were able to show in a paper that we published in Circulation many years ago that riboflavin, giving the cofactor in a targeted way to people with the double copy of the gene, so the TT genotype, two dodgy copies of the gene, resulted in very significant lowering of homocysteine. So this was the first evidence that this was a novel gene-nutrient interaction, giving riboflavin to CCs, given them to heterozygous, no effect on homocysteine. It was only when you gave riboflavin to TTs that you saw a very significant effect. Around this time, GWAS studies were showing that MTHFR was one of 8 genes linked with blood pressure, and so we began to ask the same questions using blood pressure as the phenotype, looking at targeted intervention with riboflavin with blood pressure as our endpoint, and I just want to share those with you as the final part of my talk today. We recruited cardiovascular patients from a local hospital. We pre-screened several hundred of them to find those that had the TT genotype, and then we age and sex matched the TTs to similar numbers of CCs and CTs, and we looked at blood pressure and found a very powerful genotype effect as you went from no 1 or 2 copies of the variant. Blood pressure, both systolic and diastolic, increased. But actually of greater interest when we gave riboflavin for just 16 weeks, and this was a NARDA level of riboflavin, this wasn't a pharmacological level, we got profound lowering, just like the homocysteine, very gene-specific stuff, no change in blood pressure in the CCs or the CTs, but very marked blood pressure lowering that was not just of statistical significance, but important clinical significance. This is a blood pressure lowering of 13 millimeters of mercury in systolic blood pressure. We went on to be able to show more recently that this just wasn't a quirk of these high-risk cardiovascular patients. I should have said that those were premature cardiovascular disease risk. The riboflavin story also worked in people generally with hypertension, but no overt CVD. And then there was a few unanswered questions, and in my last couple of slides I'm going to share results that are hot off the press. Well, they're actually not off the press because they haven't been published yet, but we've just found them on data that we have on nearly 6,000 older Irish adults. And again, it was one of the sister projects funded by the joint Irish Nutrigenomic Initiative that I mentioned, funded by the Irish Department of Agriculture. And we were able to ask because we had huge numbers and we had the relevant data. We had blood pressure data, we had genotype data, and importantly, micronutrient data, both intake and biomarker status. And we were able to answer important questions that we hadn't been able to address in previous studies. We were able to address how important MTHFR genotype was relative to other factors that predisposed to blood pressure. We were able to question whether drugs, antihypertensive drugs, were important or not, because in all our previous studies, the people were already on antihypertensives. I should have said that. We didn't change their antihypertensives. The riboflavin effect was over and above the antihypertensives. But particularly we were interested in this question: did the MTHFR genotype increase the risk of developing hypertension? And so you're looking at over 5,000 data on Irish adults. We looked at these with and without people on antihypertensives, and the picture is the same. So antihypertensive usage doesn't change this picture. And really what you can see, the important thing is that the genotype, the phenotype is there. These are aged 18 to 90-year-olds, and the phenotype is there from age 18 on. Profound elevation in systolic and diastolic blood pressure in people who have the TT genotype in this polymorphism. The data are just shrunk down into deciles for presentation purposes, but the phenotype is strongly there. In fact, by age about 65 years, you stop being able to see the genotype effect, presumably because of other age-related factors that predispose to blood pressure catching up. We just a quick look at changing around, trying to look to speculatively ask, well, among this sample, if you had good versus low B vitamin status, did that change? And so it's maybe a wee bit easier to see on the diastolic plot below. It's systolic upper plot, diastolic down here. So for both, the broken line is showing what happens if you've optimal status. So the filled in line, the red is the TTs, and really they don't know their genotype, they don't know it's predisposing to blood pressure, but if they happen to have a good diet that has rich source of riboflavin, then hypothetically they'll be down in this category where they don't show much of a phenotype. So it is possible to modulate this. We think this matters because of the huge risk factor that blood pressure carries, increasing with mortality. One meta-analysis estimated that a 2 millimeter decrease in systolic blood pressure could lower the risk of stroke by 10%. So we think it's hugely important. We think the impact might be very important of this genotype effect and its responsiveness to riboflavin. What are the applications? Optimized riboflavin could be achieved through supplementation, fortification either of specific foods or mandatory fortification, as in the United States, or indeed dietary rich sources. These could be achieved without any adverse effects because it's low levels of micronutrient, it's not pharmacological levels. Some evidence there that riboflavin might be a particular problem in the UK population generally. So we think this is important. I've just lined up the genotype-specific effect of riboflavin against other dietary measures to try and see the impact. And we get somewhere between, depending on the age, we get the best effect in younger adults, but depending on the age, targeted intervention with riboflavin can lower systolic blood pressure by somewhere between 6 and 13 millimeters of mercury. You can see how the other dietary measures measure up. Weight loss can give you substantial decreases, but the biggest problem is you need substantial weight loss and you need to sustain it, and we all know how difficult that is. So really, our work on riboflavin so far, to summarise, Chairman, the work on riboflavin is we show that this genotype increases the risk of developing hypertension. That's really important because our work so far showed us it could treat it, but this can prevent, this genotype seems to be there in terms of someone's predisposition to developing it. Riboflavin can play an important role in the prevention of hypertension, specifically in the 1 in 10 Irish adults and greater numbers in some subpopulations, and it's independent of antihypertensive therapy, and there's the potential for a personalised approach to managing hypertension. I'm summarising the key talks then. We asked 3 quick questions. Can an optimal folate status be achieved through natural food sources alone? No. Is there evidence demonstrating beneficial health effects of fortification and supplementation with folic acid? Yes. It's a proven effect in NTD, a probable effect in stroke prevention. Is there a role for personalised nutrition in the case of folate and related B vitamins? And it's a yes. And just to thank my collaborators and all our very generous funders for the work that we're doing in folate and the related B vitamins. And a particular thanks to my lifelong mentor, originally my supervisor and colleague, John Scott, a guru in the folate field who I had the great pleasure of working with right up until his death just this time last year. Thank you very much for your attention.